Hi, happy morning, children. Hope you all are doing good. Thank you for inviting me into your home. I feel extremely happy to be with you after more than 1.5 months. Hope you are staying safe. My greetings to all your parents too. This class is really going to be a very interesting class. A new adventure in your life. Mostly when you sit and learn, you'll be in a classroom with a blackboard and teacher will be there monitoring you and you all sit together in bench rows. But now it's a new experience where the classroom is brought into your home. So enjoy learning, gain knowledge and make use of this time. I'm going to start with the first lesson, children. The first lesson is reproduction in plants. It's unit 6. The lesson topic is asexual and sexual reproduction in plants. This particular lesson consists of 10 topics. It starts with asexual reproduction, vegetative reproduction, sexual reproduction, then comes pre-fertilization events, fertilization events, then post-fertilization events, apomix, polyembryony and parthenocarpy. So these are the topics that we are going to discuss in our classes. And today I'll be going with a few topics. That is the first 3 with the introduction, the asexual reproduction and the vegetative reproduction. So before going into the lesson, let's see few of our scientists, few of the embryologists who has marked their era in the discovery of things. The first comes Nehemiah Grew. He was the one who mentioned stamen as the male organ of a flower. The next person is R.J. Camarius. He described the structure of flower, anther, pollen and the ovule. Third one is J.G. Coldreauter. Gave a detailed account of the importance of insects in pollination. The next one is G.B. Amici. He discovered the pollen tube. Then in 1848, Hofmeister described the structure of pollen tetroid. See, the pollen grain you may have learned in lower classes, and this pollen grain is made up of 4 units, and this was found out by this man. And few more scientists are there which you can see in the slides and you can read them on your own. This scientist names are very important, children. Whenever you get into a lesson, the lesson begins with the name of the scientist, name of the person who has worked in that particular chapter, particular concept, particular lesson. And those names you need to learn and what are the contributions they have made, because you may be getting that in your 1-mark questions, in your multiple choice. If you don't learn this, you may be losing your mark. So always concentrate in the names of the scientist. Learning all these 12 together may not be possible in a single time, but every day you can learn 2 names, 2 years and their contribution to the field of science. Let's move to the definition of this lesson. The definition is a reproduction. What is a reproduction? It is a vital process for the existence of the species, and it also brings about suitable change through variation in the offspring for their survival on earth. So here the definition, I noted 4 important points which I am going to discuss with you. Vital process. Vital process means a very important process. We find many important physiological activity taking place, and one of such important one is a reproduction. Why do reproduction is given such an important thing? The whole universe is made up of living things and non-living things. Living things cannot survive without the help of the non-abiotic factors. If a plant needs to grow, the plant needs sunlight. it needs water, it needs air. So this biotic plant depends upon the abiotic factors. Now this plant is growing. This plant grows not only for a beauty, but it has energy in it which is taken by the herbivorous. The herbivorous in turn are eaten by the carnivorous. The carnivorous are in turn eaten by the top carnivorous. So when you see this line, we find that the number of plants are decreasing. The number of animals are decreasing. So there is no use in them growing every day. But if we want to maintain the complete cycle, the plant which was eaten by the animal need to grow again. A new plant has to be grown. Only then the next herbivore will be able to survive, because those herbivores eats upon this plant. So for the survival of the entire entity, entire universe, every plant and animal need to undergo the vital process called the reproduction for the existence of the species. If their family, if their groups, if their organisms need to be living for a long time, this reproduction has to take place. So that is a very important thing that is mentioned in the definition. Now we'll move on to the types of reproduction. Reproduction is basically divided into two types. You would have learned in your lower classes. One is the asexual reproduction, the other one is the sexual reproduction. Let me say the difference between the asexual and the sexual. In asexual reproduction, you need not have the gametes. Gametes are very important. If you take an organism, the organism may be of two types. One is a male and the female. The male organism produces a male gamete. The female organism produces a female gamete. This male and the female gamete, they fuse. We call that process called the fertilization. So the production of the gametes and the fusion of the gametes does not take place in the asexual reproduction, because they don't have gamete in them. They are called asexual reproduction. With the vice versa, what would be sexual reproduction? They have the production of gametes. We call that process as the gametogenesis. And the next step they do is they fuse. The male gamete and the female gamete, they fuse, which we call it as the fertilization. So basic difference between asexual and sexual reproduction is, in asexual there is no formation of gametes, there is no fusion of gametes. If you take sexual reproduction, there is production of gametes and there is fusion of gametes. So this is the basic difference. Let's move into the second topic, that is asexual reproduction. Asexual reproduction, as I mentioned before, there is no production of gametes. As a result, there is no more fusion of the gametes. This asexual reproduction does not involve two organisms. It can be possible if there is even a single organism present. This asexual reproduction is of different types. In lower plants, we have conidia. Conidia in penicillium, budding in hydra, fragmentation in spirogyra, production of gemme in markensia, regeneration in planaria. I hope you all have seen the pictures and you would have got an idea. So these organisms, they don't have a fusion. They just reproduce by breaking up their parts of any part of their body. Those broken parts have the ability to grow into a new organisms, which we call it as the asexual reproduction. In plants, this asexual reproduction is called by a peculiar name called vegetative propagation. So asexual in plants is known by a name called the vegetative propagation. For this propagation, the plants needs buds. Do you know what is a bud? A small leaf-like structure which grows in between the leaves, the axil of the leaf. A small green color tissue, leaf-like tissue, which we call it as a bud. This bud grow and develop into a new plant. What a wonder it is, no? Usually the buds blooms into a flower, or bud just blooms into a leaf. But this bud, in certain plants, they have the ability to grow into a new plant. We call such buds as reproductive propagules or diospores. What is the name given for it? Reproductive propagule or diospore. Now this vegetative propagation, before moving into it, let's see what are the types of vegetative propagation, that is asexual reproduction. It is basically divided into two types. One is the natural method, the other one is the artificial method. You can see that in the slide, and the first one is a natural method. This natural method is very common, which occurs on its own in the nature, whereas the artificial method, which we have to induce doing, we have to make an artificial method to produce a new plant. Let's start with the natural method. Vegetative propagation by natural method is divided into three types. I told you before itself. This vegetative propagation is basically by the buds. So buds are in the roots. Those buds in the roots help to grow into a new plant. Buds are present in the stem, and those buds in the stems helps to grow into a new plant. Buds are present in the leaves. Those buds in the leaves helps to grow into a new plant. Now the names are given for these buds. The buds that is present in the root is called the adventitious bud. Those present in the stem is called the axillary bud. Those present on the leaves is called the epiphyllous bud. I'll show few slides for you showing the example for the vegetative reproduction. You can see the slides with the names given over here. In the root, vegetative propagation of the root, you can find Ipomia batutus and Dahlia. In the vegetative propagation of the stem, you can find Musa paradisiaca, Ginger officinalis, Solanum tuber, Bulb allium cepa, Sucker, Offset, Bulbils. So these are the methods in which the plant grow. Now in vegetative propagation in leaf, the very best example is bryophyllum. Do you know what is bryophyllum? Kattipota, kuttipodam. Okay, this leaf has got notches. In these notches you will find buds, and these buds, they help the plant to grow into a new plant. This is not actually, bryophyllum is not a plant, it is a leaf. In the leaf of the bryophyllum, down the notches you have got a bud. That bud helps in to grow into a new plant. We call such buds as the epiphyllous bud. So these are the various methods of natural vegetative propagation. Now let's move on to the next topic called the artificial method. Artificial method, we are going to induce, we are going to make plant reproduce. Okay, so this method is divided further into two types. One is conventional method, the other one is the modern method. Conventional method is further divided into cutting and grafting. And if you go to your grandma house, sometimes you may break up your hibiscus plant and take and plant it in your home. And people visiting Ooty, Kodaikanal, they take rose cuttings, a branch of the plant, and they plant it, break from those places, bring it to their home and plant it in their houses. After sometimes you can find the plant coming out of those small branch which is taken up. This is called as cutting, which can be done in hibiscus, bougainvillea, moringa. Okay, these are the examples for cutting. Now coming to grafting. What is grafting? For doing grafting, you need two pieces. One of the two plant, the plant which is in contact with the soil is called the stalk. And the other plant which we are going to introduce into that stalk is called the scion. So we need two plants. You can notice in the picture, there are two plants given there. One is connected in the soil, which we call it as the stalk. The other one, can you see the green color one, which is kept over the stalk, we call it as the scion. So grafting has two members, it needs two different members. These two plants may not be of the same species. It may be of different species. Whereas in cutting, it's only a single plant which we cut, we plant. But here in grafting, we are mixing two varieties. So grafting is divided into 5 types. Bud grafting, you can look into the picture. Approach grafting, tongue grafting, crown grafting, wedge grafting. In this, you can find the plants are not of the same type, but the two are introduced. The last topic is layering. Layering is another important conventional method. What is a layering? See, the stem of a parent plant is allowed to develop roots while still in contact. You can see a picture over here. A branch of that mother plant is bent down and a stone is kept over the branches. And the place where the stone is kept, you find roots growing by. Once root is grown and that branch which is bent is cut, so that plant grows separately, and the other one with the root, newly formed root, they grow into another plant. We call this kind of thing as a mound layering. In the second picture, you can find the branch of the stem is tied up with a wet cloth or a polythene bag, so that the connection between the xylem of the branches is stopped. And in this region, a soft soil, wet soil is coiled over, so that roots develop in this region. And the branch which is tied out of the bag, they grow into a new plant. We call it as the air layering. So these are the 2 types of layering. What are the 3 basic things we learned in the conventional method? Cutting, grafting, and layering. Now we'll move on to the next topic, that is a modern method. In this modern method is very much efficient and it is lab oriented, whereas the other things, cutting, grafting, layering, we can do it in the field. But this modern method of growing plant, it is done in the lab. What is unique about this is we take a tissue, a small piece of the cell from the plant, which we call it as the explant. Then we culture it in the conical flask with the culture medium. Then transfer it into the test tube. This grows into calyx, where the calyx are differentiated into root and the shoot. Further, they grow into a new plantlets. These plantlets, after some times, they have the ability to grow into a new plant. So this new plant which we see now is purely taken from a small tissue. A small piece of tissue is able to produce a large plant. This process is a very unique advancement in the field of science. We call it as micropropagation plant tissue culture. So these are the various methods in which plants reproduce without the use of the sex organs. Children, hope you understood these lessons, and if you have any doubts in it, you can clarify it with me with your queries to your class WhatsApp group, and you can ask me the questions and I'm ready to explain you. Hope you understand it, and I've given you certain homeworks at the last of this video. You can take up those and concentrate in it and make a very thorough preparation, and each and every topic is going to be very important for you. Thank you children for listening so patiently. We'll meet in the next class. Bye.